think I'm going to give it another. Minute or two. Are there people logged in online? Okay. I'll give people kind of another.
Yep, take it down to the wire.
I tend to be one of those people. Okay, should we get started? Hello. We're gonna get started, yeah? Is that cool? Silvestre, can you hear me okay on this mic? Yeah? Okay, thank you. The microphone is always weird to me because you can't actually hear it in this room. You just have to trust that the people on the online universe can hear it. So welcome everyone. It's my pleasure today to introduce Lilian Carswell and Michelle Staedler. Michelle is sitting down here. She's sort of the assistant and will be available for answering questions. So I'll start by introducing Lilian. So Lilian has a really interesting career trajectory. As an undergraduate at UC Berkeley, she double majored in environmental science and English. And then she sort of carried that theme through her career. So she went on to get a PhD in English in Columbia University, and the title of her thesis is Telling the Truth About Animals, Epistemology, Ethics, and Animal Minds in Melville, Darwin, Saunders, and London. And then after that, at some point came back to graduate school and did a master's at UC Santa Cruz and is now in the process of completing her PhD at UC Santa Cruz. But throughout all of that time has consistently worked in a variety of settings all around the same theme. So she served as a wildlife biology technician, biological science technician for sea turtles at the USGS in Texas, biological science technician working on sea turtles and red wolves in Florida. She's worked for U.S. Fish and Wildlife, and right now is the Southern Sea Otter Recovery and Marine Conservation Coordinator for U.S. Fish and Wildlife at UC Santa Cruz. So I just kind of love this like English and marine ecology stuff carrying throughout your career. And then I'll go ahead and introduce Michelle as well, who is a collaborator on this project. So Michelle has a BA and a master's degree from UC Santa Cruz, which is where they met doing their sea otter work. And she's held a variety of positions, and particularly in the Monterey Bay Aquarium. Started out as a research assistant and then became the Sea Otter Research Coordinator and the Sea Otter Program Manager, and has been also involved in several documentary filmmaking efforts, and currently is working for the USGS and UCSC in the Sea Otter surveys. So these are the folks you might remember a few weeks back. I gave a little blurb about they're set up at Marine Ops doing some survey work on otters, so I'm looking forward to hearing more.
Thank you so much. Can everyone hear me okay? Great. Thank you for being here. It's such a pleasure to be able to talk to you about sea otters and what we're doing in Elkhorn Slough. So I should say that this is research that is underway and that we really haven't done most of the analysis yet. So this talk is heavy on methods and very light on results, but we're happy to share with you what we do have so far and to speculate along with you with whatever questions you may have. So I know acknowledgements typically come at the end of a talk, but I'm giving them now just to emphasize that this is an extremely collaborative project and that there are actually more than 100 people have been involved so far in one way or another. And also many organizations, and not the least of which is Moss Landing Marine Labs. The small boat facility has been absolutely instrumental in the work that we've done. If we didn't have access to that, we would be in some horrible little muddy spot in the marsh, like trying to get up and down a muddy road next to the Moonglow Dairy. It would be a total nightmare. So thank you so much for your generosity. And JD, thank you for coordinating with us and being so open to us being there. It's been incredible. Like when people heard, they were like, what, electricity and water? This is amazing. Okay, so I'm going to give you a little sea otter history and outline the conservation framework in which this research is taking place. So I have kind of a dual role for the U.S. Fish and Wildlife Service. I'm in charge of helping to coordinate sea otter recovery with all the many partners that you saw on that previous slide. But I'm also doing my PhD, so I also have more esoteric interests in social behavior and things. But this is sort of the conservation framework of the work that we're doing. So the southern sea otter is one of 3 sea otter subspecies. The other subspecies are the Asian and northern sea otters, and those predictably occur in Asia, so off Japan and Russia. That's the Asian sea otter. The northern sea otter is all through Alaska, BC, and there's a reintroduced population in Washington that I'll show you a map in a second. Oh, here's the map. Handy. And then there's the southern sea otter, which historically ranged from around Oregon to midway down Baja California. So there's the terminus of the range. And here's sort of a transition area between northern and southern sea otters that we believe existed in the past. So just to orient you to this graphic here, the dark colors are existing range, so that's the reintroduced population of northern sea otters in Washington. This was not a reintroduced population. There was a remnant population that survived right around there after the fur trade, and it has slowly reclaimed this portion of range. And then San Nicolas Island, there's a reintroduced population there. All this pale orange is historical southern sea otter range. So the southern sea otter is federally protected under the Endangered Species Act and Marine Mammal Protection Act. But despite having been protected for more than a century, it still has a really limited range. And in fact, this area from Washington all the way down here to Pigeon Point, which is south of the San Francisco Bay, is devoid of sea otters, and that's actually the largest remaining gap in the worldwide sea otter range. So recovery has happened pretty well, especially in Alaska, but also off Bering Island, and then it's starting to happen in northern Japan. But we have this problem in California where we have just had very limited range expansion, and one of the primary reasons for that is white shark bite mortality, and that's especially at the range peripheries. So the edge right there, the edge right there. Shark mortality has just been increasing almost exponentially over the past 20 years, and we have not had any net range expansion. So we've had like a couple blips where they move forward briefly and then retract it again. So all of that has led to talk about reintroduction. And reintroduction has been proposed to both facilitate the expansion of the population and increase gene flow between the two subspecies which once existed, and also to restore the native marine ecosystems of which sea otters are a keystone species. And so in 2020, Congress actually directed the U.S. Fish and Wildlife Service to produce a feasibility assessment on the possibility of reintroducing sea otters to the Pacific coast of the U.S. So we released that assessment in 2022 and concluded that reintroduction was feasible, but that we needed to do more work to evaluate the socioeconomic impacts. So there are 2 super broad questions that you would consider when thinking about reintroduction, and those would be where and how. So starting with the where, estuaries have been a major focus. There's no decisions have been made of any kind, so all of this is just what people are talking about, what we think might happen. Estuaries have been a focus precisely because of the white shark bite mortality. White sharks occur all up and down the coast, and climate change seems to be bringing the juvenile sharks further north for longer periods of time. So white shark mortality just seems to be like here to stay. So estuaries are generally pretty shallow, generally too shallow for white sharks to enter. I know white sharks can enter San Francisco Bay, but there are lots and lots of shallow areas around there. And then some of the smaller estuaries, really, it would be sort of unheard of to ever see a white shark in there. So for that reason, and others, estuaries have kind of risen to the top of the list. In addition to safety from white sharks, they also provide shelter from rough waves and winds. They have an environment that's more like the human care facilities that rehab otters are raised in. I'll talk about that a little more in a second. They have some degree of physical containment, so it might help to stop dispersal, or at least impede dispersal a little bit from a reintroduction site. And there's a somewhat reduced potential for conflict with shellfish fisheries. But there are also downsides to estuaries. If they're small, and if there's a bunch of sharks right outside, they're going to reach carrying capacity pretty quickly, and then where are those otters going to go? It's sort of like an isolated pocket, and it will remain isolated, you know, maybe forever. Or at least, like, there will be a very small chance that otters will be able to get out of there and start populating other areas of the range. Also, estuaries tend to be places where humans like to recreate, do other things, build ports, build docks. So there is pretty high capacity for human disturbance. So those things raise the question, what changes are likely to occur after an estuary is fully recolonized? And what I'm really talking about is after sea otters deplete the prey resources that have accumulated during their long absence. And then what are the trade-offs between estuarine and outer coast habitats? Not just the threats, so we already talked about the threats. The sharks are really a major threat to sea otters. But you know, what about the other cumulative stressors that occur in estuaries? How do we evaluate those? How do we measure, like, how bad and how good is this place relative to another place that we could put sea otters? So then for the next question, how, it's huge, but one major gap in our knowledge about sea otters is what their social structure is like and whether social affiliation matters. Previous reintroductions have all used wild sea otters, and they've lost about 90% of the otters, both to emigration and mortality. So they didn't all die, but they either left or died, and just a tiny core remained. So that doesn't mean that that doesn't work, because all the previous translocations used wild otters, and a third of the otters alive today in the world are the result of those translocations. But you just have a high loss initially, and some of that is mortality. And so we're not living in the cowboy days anymore, so we really want to avoid that to the extent possible. So how do we do that? Is it possible that social factors are important, and is there any way that we could improve the social environment for sea otters in a reintroduction site that would make them more likely to stay? And we really don't know. So one other question that comes to mind that it'll become clear why this matters if you're not already thinking about it, are affiliated sea otters more closely related? And so that brings up the question of genetic diversity. So southern sea otters are both the most genetically distinct of the three subspecies, and they're also the most genetically impoverished of the three subspecies. So they really, like, sea otters in general have very low genetic diversity. They had the bottleneck from the fur trade. They had bottlenecks even before that that have been detected through genetic studies. But southern sea otters they have the biggest problem in that respect. And then when you add that to the factor that reintroduced populations just typically, they start small and they're typically isolated. So then you have this potential for inbreeding. And so if we find affiliation does matter, and we're like, oh gosh, okay, let's reintroduce clusters of affiliated sea otters. Well, are those otters all related, and how is that going to impact the genetics of our reintroduced population? So all these questions are in the mix and driving my research, which focuses on 3 primary questions. So the first question relates to body condition and habitat use, and that's the question of how does that change, how does that turn out in an estuary over time? Like, can an estuary sustain a population over time? What kind of numbers can we expect? What sort of densities can we expect? And what happens when they start to be strapped for food resources? What changes? Do they use more marginal habitats? Do they use more marginal food? Do they just leave? Do they die? What happens? So there was a study in Elkhorn Slough 2013 to 2015, which it provides a really excellent baseline for us to compare the current situation to. And that study seemed to be occurring right at the time that sea otters were reaching carrying capacity within the slough. And carrying capacity for sea otters is pretty much determined by prey. Like, other things matter, yes, but prey is the main determinant. And of course, prey populations, they fluctuate. They have, recruitment events, they have depletion events. So I'm not implying that there is just one carrying capacity and that's it. I'm just saying we tend to see, when sea otters deplete the stores of prey that have accumulated in their absence, we tend to see certain patterns. So the expectation here would be that body condition would decrease, and body condition is basically sort of a formula, length, weight, whatever, like how robust is that otter? A simple way to say it was like, is that otter fat enough, or is that otter super skinny and on the edge? And then the other prediction would be that habitat use would increase to include areas that aren't necessarily the preferred areas, just because of the competition for space. In relation to chronic stress, my main question is, how does chronic stress vary with exposure to stressors? And so we don't know anything about chronic stress in sea otters. No one has ever tried to measure it, but in other mammals, people have used hair as the matrix that they then extract cortisol from to understand the chronic stress levels of these different mammals. So I'm attempting to do this with sea otters, which is kind of rough because sea otters have the most dense hair of any mammal, and you have to separate the guard hairs from the under hairs because they incorporate cortisol differently. So that's a total nightmare. So I'm also exploring whiskers in the hope that whiskers could be a replacement because they're much easier to handle. So the expectation here is that cortisol levels will represent a cumulative response to all these stressors, which include food limitation, disease, whether there's predators present, anthropogenic disturbance, and social dynamics. And the social factors can both reduce the stress for animals, but they could potentially increase the stress. So like if you're a young male and you're trying to become a territorial male and this other male is beating you up and driving you away, that's a source of stress too. So the social factors, there's kind of like a, both positive and negative. So it's, obviously difficult to tease apart all these factors, but that is something that I hope to make some progress in doing. And then the third questions relate to social structure. So first of all, do sea otters even exhibit social preferences, or are they just aggregations of animals? And past researchers have been really heavily focused on ecology, really very little on social behavior, social affiliation. And there are assertions in many papers saying, oh, they're just aggregations of otters, like you've seen one, you've seen them all, or like they're mix and match. But does it matter? So we want to know. One of the main points of this study is, does it matter who you're with? Or does it just matter that you're with some warm bodies, And then the next question is, if those social affiliations are real, are those otters more closely related to each other than to other conspecifics in the population? And so the expectation that I have is that sea otters will indeed form durable social bonds. And I do expect that females will probably be more closely related to each other if they're socially affiliated, but that socially affiliated males will not be. And that expectation is just driven by something that we already know, which is that young males are pretty much forced to disperse from their natal area, whereas females have a choice. They can stay, or sometimes they go, but they're not driven out by territorial males. So, methods. So staging ourselves at the MLML boat facility, we then take our boats out and try to capture otters with these nets. Now, this is not the standard method of catching otters. The standard method is using divers with rebreathers and ambushing them from below with these cool trap things with a drawstring, and that is a super efficient way to catch otters, but it requires clear water, and it requires water that's deep enough for divers to stage in, and it requires waters that aren't like massively, flowing in and out with big tides. So all of those, that's all just impossible in the slough. So in the slough, we have to use these tangle nets, and otters, unfortunately, are intelligent creatures. They're aware, they examine things, they analyze the situation. So over the 3 years, we have noticed a marked decrease in our ability to catch otters as they catch on. But these are the methods. So we set the tangle net either in the main portion of the slough, so this is in the main channel of the slough, or we set them across these tidal creeks, which are like the in and out, the entrances and exits of these marsh areas where sea otters like to haul out during high tide. Once we get an otter in the net, then we use these dip nets to scoop them up and secure them, because if they're in the tangle net, they can sometimes get out. So we scoop them in these dip nets and bring them on board, and then we drop them into these wooden capture boxes. And these capture boxes are custom made for sea otters, and they've been used for decades, and they're really the tried and true method. And then when they're in the capture box, then we have this elaborate little method of getting the net off the animal that I can explain to you if you want to know. After that, the otters are transported to your facility, where we have, the CDFW vet lab right here, where they process all the blood. We have these tables set up, lots of vets around. There's Michelle. She's the queen of the shore. She organizes everything that happens there. So we sedate the otter, and then we sample the crap out of it. And so it's not just for my study, it's for everyone's study. Like before we do a capture, we send out an email to all the other partners that we've basically ever had, or anyone we know who's interested in samples, and we're like, hey, we're catching otters, do you want XYZ? Send us your tubes, send us your instructions. So we're collecting samples for a whole bunch of studies, because it's hard to get an otter on the table, and both for us and for the otter, we don't want to waste this opportunity. So we do all the sampling, we record everything, and then we put some flipper tags on. Now we're still in the dark ages with sea otters. You can ask me why in the Q&A session. These are just plain plastic tags, cattle tags, the cattle ear tags that we put on their flippers. But they're really surprisingly effective, especially in the slough. So we're trying to tag as many otters as possible so we can recognize all the individuals, because we want to know who's with who. That's the main thing, and we also want to know who's doing great foraging day after day and who's doing terribly foraging day after day, who's in this area that's exposed to high anthropogenic disturbance, who's always in this other area that never has any anthropogenic disturbance. We want to understand all those things so that we can start to tease out those factors of what stressors are affecting particular animals. So this is how we place the tags. We always talk about the right flipper first. And so we have those colors that we use, particular colors, and then we have these positions. And so here the tail is in the center, and so the 1-2 position are the digits closest to, here's the tail, 1-2 position closest to the tail, the 4-5 position furthest from the tail. And so by knowing which color is on which flipper in which position, we know what animal that is. So then we use this cheat sheet, and all these, the right tag color is the first color you see there. So we're like, okay, we saw an animal with red on the right. You can't see the right flipper too well, but and red 1-2 on the left. So we go, I can't even totally read it, but yeah, there we go. Red 1-2, red 1-2. Oh, that's Ruby. Here's another example. This is purple in the 4-5 position. Purple 4-5, purple 4-5. So here we go to our handy cheat sheet. Here's a purple 4-5. Oh, there's the other purple 4-5. Oh, that's the male's warty. So that's how we identify who they are. And if you're ever out in the slough and you want to know who you're seeing, you can take note of the colors and positions and ask us, or we can provide you with our handy cheat sheet today, and you can perform recites for us at your will. Oh, look, there are the answers. Okay, so after we put the tags in, oh yeah, and we also put a pit tag in so that when they lose their flipper tags, which they do, if we ever recapture them or if they turn up dead, we can scan them and know who it was. So after all that, then we reverse sedation. We have vets keeping careful watch on the otter to make sure that the otter wakes up properly. And as soon as that otter wakes up, they're like, wah! And they slam the door shut. And then that otter goes on a boat and is released. This is what a release looks like. We let them take their time. So that is a male sea otter. He's territorial over by the hummingbird area. And we take care to release otters in really pretty much the location where we capture them. And that's especially important for territorial males because we absolutely don't want to drop them in some other male's territory and have them get beat up as a result of it. So then after all that, that's like the more exciting part, according to some. And then the rest of the year, we do recites and foraging observations. So this is over in the Parsons Slough, Five Fingers area that we call Avila. We use these Questar telescopes. Questar telescopes for our observations from land. We also do observations from the boat. And when we do a recite, we collect data on the otter's position. We just look on Google Maps and drop a pin where they are, collect the GPS coordinates, record the number and identity of any other otters that it's with, if those otters are tagged, and we hope they are. We record their reproductive status. Do they have a small pup, large pup? Are they territorial? And also their behavior. And then for foraging, when we're doing those observations, which are almost always from shore using a Questar, we record the duration of each dive and the surface time. We record whether the dive was successful, what the prey was, what the number and type of the prey items were, and the prey handling time. So that's all the field work, and then there's lots of lab stuff to do. This is the separating the guard hairs from the under hairs, which is really time-consuming and terrible, and that's why I have a furrowed brow in that picture. Extract the cortisol, and then just to even understand how cortisol in hair works in otters, we have to do a bunch of groundwork like evaluating, do body areas make a difference? Does hair type make a difference? Does storage and treatment make a difference? How do hairs and whiskers compare? So that's sort of like the basic grunt work that I have to do just even to interpret the cortisol that we're finding. Then to evaluate the strength of the stressors, I'll be using the calculated body condition index and the foraging efficiency as a metric for nutritional stress. I'll be looking at the otter's home range and overlay with human activity levels to get sort of an index of anthropogenic disturbance that the animal's exposed to. And then use some magic math to try to tease apart all these stressors. For social structure, I'll be using the Recite database to evaluate the affinities between individuals by creating a pairwise affinity matrix, meaning like is this otter always found with this other otter, or what proportion of the time are they found with this other otter? Or are they loners? Are they never found with any others? Then also, we'll be extracting DNA and doing genomic analysis to try to determine relatedness and comparing that to the levels of affiliation. So results, we have exactly 100 study otters right now. 3 adult females and 1 adult male were actually captured in that previous study I mentioned. And so they've been around this whole time. They'd lost their tags. They were off our radar. We recaptured them, read the PID tag, and we're like, oh my God, this is so-and-so from back in 2013, which was just super cool because you have that entire database of their activity back then. And you know that they lived this whole time. They're still in the slough, and then we can compare what they're doing now to what they did then. And now they're, you know, like an old man or an old lady. You know, we could see how they are relative to when they were young. We have 2 female pups and 3 male pups that we have been able to tag that were captured with their mother. And so these are super important because these are known mother-pup relationships, which is really like one of the things I really want to understand is, you know, how often do those female pups really hang around and do they interact with their mother time after time through the years, or are they just like, you know, you're a stranger to me now, I don't even know you. So we're going to get a lot of that information from the genomic analysis, but to have the actual tagged pairs to validate that is really important. Then occasionally we catch moms and pups where the pups are too small to flipper tag because they need to be a certain size to be able to safely put those tags on them. So those pups get pit tags. They don't get flipper tags, so they're kind of invisible to us. But if we catch them in some future study, or you know, if we catch one this year that we pit tagged 2 years ago, we can be like, oh my God, this is the pup of so-and-so, which would be very exciting. So in terms of recites, we have over 7,000 recites so far. We're currently following about 55 to 65 individuals regularly. And so you're probably like, well, you have 100 otters. How come you're only following 55 to 65? That sounds pretty bad. But some of them have died. Some of them have left. Some of them have chewed their flipper tags off and are under the radar. So we're doing our best. We do try to do recites occasionally outside the slough, but it's just such a time commitment for such a low reward. It's really hard to you know, it's hard to find the time to do that, and this is where we really would like some better tags that aren't just stupid plastic tags. So again, ask me about that later. Okay, so we've had some mortality, and when we have a mortality, we have to go get the carcass if we can. So here's, Michelle and I made an attempt. We were like, oh, there's a carcass. We'll just jump out of the boat and go get it. And we got hopelessly mired in this like quicksand. We thought we were gonna die. It was like, Get Smart, if you've ever seen that. Like, there was so much quicksand on TV back in the day. Anyway, it was just like that. I was like, oh my God, it's real. We're gonna die. And then Ron, who, you know, he's an elderly gentleman, had to come save us. He was like, get back in the boat, ladies. I'll show you how it's done. And he like crawls out there on all fours with this box top in front of him. And he's like perfectly able to navigate the mud. And he got the carcass. So good for Ron. So then when we get the carcass back, then we bring it over to CDFW, and this is Dr. Melissa Miller. She's the main pathologist, well, she's the pathologist over there, and then other people are also trained to do the necropsies, and they try to tell us the cause of death. And so far we know that 4 were for sure caused by shark bite. 1 was caused by trauma that could have either been a shark or a boat prop on the head. Really horrible, sad case. 2 were related to health, and 6 were undetermined. And often it's undetermined if we can't get the carcass fast enough, because then it's like full of holes from scavengers. We're like, is this a shark hole? Is it a scavenger hole? We don't know. So we've had a lot of pups born to our study, females. 51 pups have been born, and 7 were with their mom at the time of capture. About half of them don't make it. 29 have been successful so far, 22 were unsuccessful, and 7 are currently with their mom. So, as I mentioned, we're also watching foraging, and so we've seen some pretty interesting changes. So what you see here is 2013 is in the blue from that previous study, and then 2023 and 2024 are the 2 colors here. So we were really pretty surprised in 2023 to see such low clam representation and really a huge amount of fat innkeepers. This past year, it seems like it's gone a little bit back more to normal patterns, but it's really like clams and worms are the main things, and then to some extent crabs, to some extent these like shore crab types, not the super tasty raw crabs, and then a little bit of mussels where there's hard substrate. When you watch otters a lot, as we do, you see some super cool things. So these are unusual, so please don't go tell people you saw a picture of an otter eating fish, and you know otters eat fish, and so when they say otters eat shellfish, they're liars. This is very unusual, but super cool. So in Alaska, they eat finfish a little more. In California, it's quite rare to see them eat fish. But this is one of our study otters, Persephone. She's a total loner. We didn't see her for the first 8 months of our study after we captured her. We thought she was dead. That's how she got the name Persephone, because all of a sudden she came back to life from Hades. This is why it's good to be an English major before you enter science, because you have good names for your animals. And so here she is eating long john mudsuckers. That's the best we could do for ID. We did ask Pete Ramundi and Giacomo at UCSC, but if you have a better ID, let us know, and we'll tell them that you said that they were wrong. We've also seen several events of sea otters eating thornback rays. One of our tagged otters stole one. Michelle witnessed this. She can tell you about the whole event. She witnessed him stealing it from a female who had acquired it. And then we've seen untagged otters multiple times eating these thornback rays. We don't know how many otters are involved in this, if it's just the one, if it's multiple. But anyway, pretty cool. And then, even more weird, one of our study otters, I thought about including the video, but I didn't. One of our study otters was sort of climbing all over and ripping the fur off this decomposing sea lion carcass. And Michelle witnessed this whole thing. I was on shore, but she was right next to it. And at the beginning, the sea lion looked like a normal dead sea lion. And at the end, the sea lion had no skin on its face, and its eyeballs were gone. So that's what these otters are doing out there. Oh, and then, yeah, there's the grebe killing. Have you heard about that? Yeah, the birderer, yeah. So one of our otters, she's lost her tags or disappeared or something. She's the most charismatic of all our otters. It was Ruby, the 2 reds. She was a birderer. And another of our study otters tried to fight her for the bird carcass. Anyway, crazy stuff is happening out there. But it's not normal. Normally, they're eating worms and clams. Okay, just remember that. Okay, so this is a comparison of body condition. So this is the Elkhorn Slough 2022 study. 2022 means they were caught in the very first year of our study, 2022. So we measured their body condition when they were on the table. So I haven't updated it with our other numbers yet. But this is how it compares to Elkhorn Slough in 2013. So you can see there's been a significant decrease in body condition. It's also significantly lower than San Nicolas Island. So San Nicolas Island was a translocation I mentioned. Back in, let's see, what year was that? What? No, San Nic. Well, yeah, San Nic started in '89, but the study was done in the mid-2000s. Anyway, so that was when the population at Sandik was still quite small, very small with respect to the carrying capacity of the island. So those animals were not food limited at all, and you can see they have this great body condition. They're fat and happy over there. And then Monterey has been occupied for a pretty damn long time, and then Big Sur has been occupied since the beginning by the remnant colony. So that's the oldest occupied area. So you see this very predictable pattern. The oldest occupied area has the lowest body condition. Next up is Monterey. And so these are, the 2022 was kind of similar to Monterey. So starting to look like a population that's at carrying capacity. And then there was a comparison to Santa Barbara Channel, not significant. There were reasons for that, too complex to get into right now. Okay, so in terms of habitat use, again, it's pretty interesting because we've seen some changes just year to year. Based on the first year of our resites, we had a lot of animals in the sort of wildlife area by those 2 wildlife lookout platforms. A lot of animals in the Seal Bend area. Those tended to be juvenile males and juvenile females, and then adult males that were not trying to hold territories or weren't successful. So that's sort of considered like the bachelor or the mostly male group. And then a lot of animals, really a lot of animals using the Yampa Marsh area tended to be adult females with pups, and then animals using the Avila area. So we've seen some changes, the most significant of which is that Seal Bend group is completely gone. So all those juvenile, bachelor, whatevers went offshore, and we've been retrieving their poor shark-bitten carcasses as they trickle in. Some of them are probably surviving okay, but we've lost track of many of those otters. Okay, so here are the social affiliation results. These are preliminary as well, but just to orient you to this, the circles are individuals, and that's their study number inside. The lines indicate their pairwise affinity with a particular otter. So if you see like this otter and this otter, they have a really thick line between them. That's like they were seen together a whole bunch of times, like a high proportion of times. If they have a really thin line, they were rarely, they were seen together once. If there's no line, they were never ever seen together. So from this, we can do some cluster algorithm stuff and figure out, and the colors represent clusters, and then we can compare those to geographic areas. So there was a distinct Seal Bend group, a distinct Yampa group, a distinct Avila group. And so that's super interesting, but then even more interesting over time is, do those affiliations hold even when those otters start using different areas? Like, do those 2 otters who have a good affiliation that you always saw in Avila, do they start showing up together in the wildlife area? That's super interesting, and we shall see. So that is pretty much it. Our ongoing and future work includes finishing up our captures. We have 6 more capture days this November, and then that's it for the annual capture events. It is my ardent hope that we will periodically do captures again and maintain some kind of continuity to be able to understand the long-term fate of some of these animals. But for this particular project, this is the last year of systematic annual captures. We'll be continuing to do our resites and foraging observations through 2026. We'll be doing hair and whisker cortisol extraction, DNA processing, and the genomic analyses, and all the other data analysis. And we welcome volunteers. So should you wish to do resites for this study, we would be happy to train you. So let us know. And Michelle is a wonderful teacher, so I'll refer you to her. And that's it. We're happy to take questions, and feel free to ask Michelle questions too, because she is the field guru. Thanks.
I'm going to run the mic around, so let me know if you have questions.
observation, you're investing 6 years of your life in this study? Well, I was going to compare it to, it's very similar to Moss Landing master's thesis time frame. So I was just gonna
Yeah,
say,
come to
there's that
UC
similarity.
Santa Cruz. You can get a PhD for the same amount of work that you're throwing away here.
I object to that.
I'm kidding. But just think of the quality you're getting here, and it's just like it's shoddy over there at UC Santa Cruz. Can't believe they let some of us in that school.
So the actual question is, what percentage of the local population have you handled, and do you have tagged? Your 100 is what proportion?
Well, that is a great question. So initially, there's about 125 otters in Elkhorn Slough, but it kind of fluctuates. Sometimes it's like a ghost town, and sometimes it's just like, oh my God, where do all these otters come from? So I thought when we caught, 60 something the first year, I was like, oh my God, this is so exciting. We're tagging such a large proportion of the population. And then when we did captures year 2, I was like, this is gold. We're gonna have every otter tagged. And then there's just tons of untagged otters around. And a lot of our tagged otters are nowhere to be seen. So there is exchange happening for sure. So there's recruitment of new otters. There's otters flowing in probably from elsewhere, and some of our otters are leaving. So right now it seems like a low proportion, relatively low proportion is tagged. I don't know, Michelle, what your impression is, but I would say like 40% or 50% is tagged of the otters we see every day, even though we've tagged 100 otters and we think the population of Elkhorn Slough is like 125-ish. So there's exchange happening. We want to tag every single otter, but I know, I'm hungry for it.
I know you don't have full stats on your social dynamics for like the last couple of years, but have you noticed that there are groups of otters that all seem to be hanging out, like on that first main stretch just past the second overlook? There's always a raft of otters hanging out in that eelgrass bed. Are they like all friends? Are they all hanging out together?
They seem to be like, so we have these territorial males who seem to have those groups, and unfortunately one of our males lost his tags, and so we don't know if he's still the territorial male of that area. But so like in the wildlife area, just beyond the Wildlife East platform, just east of it, there's that big group that always hangs out. That used to be one of our study otters, Rory, Royal Blue Orange Rory, get it? He was the king there, and there was a pretty consistent group of otters there. And then we have another territorial male, Chip, who's on the south side, and his group is getting bigger and bigger. Lucky devil. And yeah, they're consistent. They're otters we expect to see there. And then in Avila is so consistent. Like, we just call them the Avila gang. Like, and if we ever see someone new there, we're like, whoa. And if we ever don't see them there, we're like, oh my God, are you okay? So yeah, we definitely see clusters. They're otters we expect to see in a group in a particular place. I don't know so much about, are those affiliated otters showing up together somewhere else? And Michelle, I don't know, do you have impressions of these things too? Would you say that, do you feel like we really are seeing clusters of socially affiliated otters? And do you think that we have seen affiliated otters showing up somewhere else together?
There's not a whole lot of places that they can show up together in the slough, so most of them are in those 3 or 4 spots that we showed on the graph tape in the slideshow. So the ones that do hang out seem to be, and that keep their tags, seem to be the ones that are staying together. Like with Avila, we have, 4 or 5 that are always there when we go, and the other spots seem to have the same animals at the same all together too, but occasionally you'll see 1 or 2 of them sort of venture off and go all by themselves, and you wonder why. So that's another thing. And then something you didn't mention is the loners, that we have several females that we've seen that don't join up with any group, and they're way back up in Kirby Park and way beyond there. And then we have a male who is on one side of the railroad tracks. He crosses the railroad tracks to get to the marsh, and he's been staying back there on his own. So we do have loners, and we have group people that join groups and hang out together. Or otters, I should say.
So sometimes when I'm out at the beach, I'll see otters in the surf. And so I was wondering, are those males that have been kicked out? And because why would they go out there if they know they're going to get bit by a shark? Or do they even know? And do more males get bit by sharks than females? Because the females maybe get to stay. Like you said, they get a choice, and the males don't. So I was just kind of curious on that.
Yeah, so Michelle can probably weigh in on this too, but I think, you know, Elkhorn Slough is a pretty densely occupied population, and it did seem for a while there that the prey might be depleted and that there was probably much more abundant prey offshore. In fact, one of our dead otters had a belly full of crabs, and we weren't really seeing much or any, much crab eating at all inside the slough. So I would think that the food resources are just better out there, especially because the otters are at such low densities now because they keep getting whacked by sharks. So food is not limiting them out there. It's sharks that are limiting them. But then we don't know. There's differing opinions among otter researchers about whether otters know about sharks. Some people think they have no clue because sharks are ambush predators, and they think the otters are getting whacked while they're foraging, which is usually independent. They're not around other otters. Other researchers think that otters are not stupid. They are observant, and they have the ability to recognize these things. So we don't actually know. Michelle, do you want to answer that? Yeah?
So a follow-up question on the sharks, I suppose. The general theory, say, or the general thought is that the white sharks are the ones doing a lot of the predation on the otters, but certainly there's a lot of other big pelagic sharks in Monterey Bay. Have there been a lot of analyses on your shark-bitten otters to see if it is a white shark? Have you had done analysis on bite shape and like tooth imprints? Can you confirm it's a white shark and not a seven-gill or a, prickly maybe?
So that's a great question. Michelle, do you want to take this?
Yeah, actually, there's been a lot of work done and a paper published on that too. I can't think of the name, I should, but a lot of the times when they're doing the shark or the necropsies, they will find the shark teeth in the otters, and so they've taken those out and given them to people to identify. And then they've also measured, in a lot of cases, you can see the whole mouth pattern where the bites are, and they've measured that and trying to figure out which shark it is, how big it is it a 6-footer, is it a 4-footer? So yeah, they've been making sure that they know which shark it is that's getting the otters.
Great talk, first of all. So when you capture the mom and pup and then you release them, are there any instances that impact their bond or relationship at all? Because I know there are some animals that once you touch their babies, they will reject them or they don't really like them anymore and they treat them a certain way. So does that ever impact their relationship?
That is an excellent question. So we're always very afraid of impacting their relationship. Sea otter moms are extremely devoted to their babies, and some will just scream for hours and hours and maybe even days looking for their pup. They sort of do this crazy sound to locate each other. So when we're capturing them, we obviously try to catch the mom and pup in the net at the same time. If we catch the mom but not the pup, or if we catch the pup but not the mom, we have to release the other one, and then we watch to make sure they reunite. So they're very likely to reunite because the mom really, really cares about that pup and wants to be near it and will call for it, and the pup will call back and they'll reunite. But there are occasions where they're just confused or freaked out and they don't reunite right away, and that's like a super stressful situation that we try to avoid, but it has happened. So once I think it was after both had been captured, brought, the mom was tagged, the pup wasn't, then they were released, and we released them in the exact same spot where they were captured in the channel leading into Yampa. But the boat, so we're there with the boat, and the channel's not that wide. We released them, and the mom went one way and the pup went the other, and we're like, how do we get them back together? And it was really tense for a while. The mom was going into the marsh yelling for her pup. The pup was swimming out. We're like, oh, God. But then eventually the mom did turn around and get the pup. There were a couple other occasions where we have released a pup, like if we just caught the one but not the other, like we caught the mom but not the pup once, and then the pup got temporarily kidnapped by this male out there, and then we were super freaking out about that, and then the mom went and rescued the pup and grabbed it back from the male. But that has happened a couple times. So we've never had a separation that ultimately separated both the mom and pup, but it's kind of more fraught than I would have expected. I just thought this bond is so strong, it's ridiculous to even think about a mom-pup separation. You see it in the permit, and it says, to avoid a mom-pup separation, do X, Y, Z. And I was like, what? That's not even possible. A mother would never leave her pup. But now that I've seen it, I do feel like there are circumstances where she doesn't want to leave the pup, but they go the wrong way, they get confused, they're lost, a male intervenes, there are things that can happen. So we, try to pay close attention and try to take whatever measures we can, and we've been lucky enough so far that we haven't had any incidents. But then, Michelle, do you want to talk about weanings that might happen?
The one thing I also want to mention is probably more concerning is when you have a pup with really, really small pup. So that's more likely going to be the time when the mom is going to abandon it, if she is going to, because the pup isn't really able to swim to her, whereas the older pups, they know how to swim, they can vocalize a lot and go right to mom. So when we're on shore and we have a mom with us, first of all, we don't capture moms with pups under, 3 weeks,
Yeah.
4 weeks?
Or we try not to.
We try not to, so we don't even try to get those. And we do have shore spotters that are telling the net catchers, okay, there's a mom with a little pup, don't let him get in the net, and they're very careful. But if we do catch one that small, when we sedate them, we always wake the pup up first, so it's vocalizing and in its box waiting for the mom to come and get it, and then they're holding on to each other when we release them together. That's the best way we can do to mitigate any kind of separation for them. But then we have had a couple of times when we caught pups that were just close to weaning age, and we put tags on mom and pup, and the next day we go out and they're separated and weaned, and the mom's mating with a male. So that would be probably more than likely the pup was old enough and the male took advantage of her situation and grabbed her to mate with, and the pup ended up getting weaned shortly after we tagged it.
So can you tell us when the next time is that you said you have 2 more sessions of tagging, so when are you doing that?
It's November 6th through 8th, right? It's not on election day, we know that.
I
So
gotcha.
people are going to be either really happy or really sad. We won't say which.
So if we need a little mental health therapy, we can come and check it out.
Yes.
Check out your otters. So the 7th and 8th and the 21st and the 22nd. 6, 7, and 8. 2021 and 22.
We could tell you for sure. I mean,
Yeah.
these are correct, but we can send you an email with the correct.
I have it in an
Yeah.
email actually, but and is it okay? I appreciate the ask for volunteers or the suggestion, but if folks can't volunteer, could they come check it out on the days that you're catching them?
Yes, so Michelle is in charge of everything that happens on shore, so you would contact Michelle and she would,
Okay. Yep.
yeah, it can be very boring. And those are the days that we think hard about the intelligence of sea otters.
Any last question before we wrap up? Okay, cool. Thank you.
Thank you.